Thank you all so much for joining us for this year's Researcher of the Year event. My name is Jenna Frick. I'm a Senior Media Relations Specialist for the Office of Communications and Public Affairs, and I'll also be moderating today's event. And with that, I will turn things over to UMB's President, Dr. Bruce Gerald.
Thank you, Jenna. It's a pleasure to be here, and I would welcome you all to this Researcher of the Year award. This entire week is Founders Week. It started Saturday evening. I think this is our 5th event of the week, and it's one that I always look forward to. So welcome to that. The Founders Week Researcher of the Year is given to an individual who has demonstrated a very sustained, impactful, socially important practice of medicine or law or social work importance to this campus, consistent with our overall mission of improving the health and well-being of mankind. That is UMB's mission. The particular investigator today went through a fairly serious, intense evaluation of her work compared to other applicants who were nominated for this position, and she was the finalist which the committee recommended and we selected. So I know that her work is impactful because I've followed it for years, and I look forward to seeing the whole picture. Okay, and with that, I'd like to welcome Dean Albert Reese, Dean of the School of Medicine, to introduce our Founders Week Researcher of the Year. Dean Reese.
Thank you, Dr. Gerald. Let me add my welcome to everyone who is here to celebrate the Researcher of the Year award in none other than our colleague and friend, Dr. Ronna Hertzano. By way of background, Dr. Hertzano is a professor of otorhinolaryngology, head and neck surgery, as well as anatomy and neurobiology. Dr. Hertzano is a scientist and a member of the School of Medicine's Institute of Genome Sciences. Before I proceed, I'd just like to recognize the environment in which Dr. Hertzano lives. She is in the Department of Otorhinolaryngology, and I want to recognize her chair, Department Chair, Dr. Ronna Taylor, who obviously makes that environment rich enough for her to flourish. The second environment, the scientific environment in which she is hosted, that is in the Institute of Genome Sciences, and the director of that institute is Dr. Claire Frazier. So we want to recognize both leaders and thank them for the rich environment that they have created in order to allow Dr. Hertzano to flourish and to do so very well. So Dr. Hertzano is both a surgeon and a scientist, and her many years of serious scholarly and distinguished research have more than qualified her for this prestigious award. Dr. Hertzano is one of the country's leading scholars and experts in the field of hearing loss. In particular, her clinical work focuses on the diagnosis and treatment of disease of the ear, with an emphasis on hearing restoration. She sees and treats patients of all ages who suffer from hereditary and non-hereditary auditory and vestibular dysfunction. While Dr. Hertzano is a gifted clinician, she spends about 60% of her time dedicated to research, where her goal is to make significant contributions towards the treatment of the same conditions she sees in her patients on a regular basis. Her research work is groundbreaking, which is evident by her active grant portfolio, which as of today tops out at $11 million extramural funds. Dr. Hertzano has published extensively in her field and has won numerous awards for her research as well as for her teaching. Our School of Medicine values include, among others, interdisciplinary collaborative research. And I often quote the African proverb that states, if you want to go fast, go alone. If you want to go far, go together. That's Dr. Hertzano. She goes with others, and as a consequence, she does go far. She regularly and naturally collaborates across the School of Medicine. Her thoughtfulness, generosity, and supportive nature make her an ideal leader of students and colleagues alike. Not only has Dr. Hertzano invested significantly in her own career, but she has a strong interest and indeed a passion and an ability in developing those of future researchers and lecturers. She's a valued mentor for many students, trainees, and faculty members alike. In the past 6 years, she has individually mentored more than 60 School of Medicine students. Dr. Hertzano is truly a remarkable colleague who happens to be a good friend of mine. She is a major asset to the School of Medicine, the university, and the scientific field. Ladies and gentlemen, it is now my proud and most delightful moment to introduce to you a friend and colleague, Dr. Ronna Hertzano, the UMB 2021 Researcher of the Year. Dr. Hertzano.
Thank you so much, Dr. Gerald and Dean Reese, for this incredibly kind introduction and for the honor of being here and standing here with you today. Thank you for my friends, colleagues, and family, parents, mom and dad, Jen, my partner, for being here with us as well. The talk is titled From Ear to Gear, a Multi-Omic Path Towards Therapeutics. But really, when I thought about it more, I thought a better name for it is the power of collaboration and the benefits of a collaborative research community. I have only one minor disclosure. And because the audience is general, I'd like to start with an introduction to hearing. And hearing is really a remarkable sense. I speak and you hear, and my voice is transmitted in air, and you're hearing with the sensory cells in fluid. The sound doesn't bounce back. Sound comes in through the auricle, hits the tympanic membrane, is focused through the three ossicles on a much smaller surface area that then overcomes the difference between air and fluid and generates a wave that goes throughout this amazing organ, the organ of hearing that is called the cochlea. In the center part of the organ of hearing, there's a swelling. The swelling is called the organ of Corti. And the main organ in the organ of Corti is called hair cells. These are not the cells that we have on top of our head. These are little cells that have projections on their top, and they move as a result of this fluid wave. These cells are incredibly sensitive, and in human and in mouse, they do not regenerate. And in zebrafish and in chick, they do, and it gives you some hints to some of the things we think about. And when we're born, we have 4 rows of these hair cells in our cochlea, in our organ of hearing, throughout its length. There are a row of inner hair cells, a single file of cells that has sensory innervation, and 3 rows of outer hair cells that just amplify and tune the sound. They're not sensory innervated to detect sound. And up to age 20, our hearing is fantastic. This is the range of frequencies we hear, up to 20,000 hertz. And these here are the range of frequencies that the syllables that we use in order to produce speech are. And you see that when we're young, our range of hearing far exceeds what we need in order to communicate with each other, which is by far the most important thing that we do with our sense of hearing. But with normal, healthy aging, not people that have a specific mutation, we lose many of these frequencies. By age 56 to 65, this is what an average hearing curve looks like, and it becomes difficult to distinguish sometimes between an F and a T and an S, even in quiet. And this is so common that you can Google, how old are my ears? Just go online, and you'll find roughly your age. By age 70, 50% of the population should be using hearing aids. And indeed, according to the World Health Organization statement from March 2021, there are 430 million worldwide that have disabling hearing loss. 34 million of them are children. And it is estimated that by 2050, over 700 million people will have disabling hearing loss. Hearing loss results from a variety of conditions, with noise and genetics playing major roles. But our treatments for hearing loss today are solely based on one thing: how much hearing loss we have. If we have a little hearing loss, and it doesn't matter if it's because of aging, a genetic mutation, an ototoxic drug, we're going to use a hearing aid. And if hearing aids are not enough to restore our capacity to communicate with each other, then we're going to use a cochlear implant. There are two things we'd like to accomplish. We would like to figure out ways to regenerate the hair cells, to reverse the damage. And the second thing, we would like to protect the ears. How wonderful would it be if the hearing that we have when we're 20 could be the same when we're 70? Are there ways that we can accomplish this goal? I will start with the first. We're trying to think about ways to approach this question. Obviously, we're one of many, many different groups, and we're part also of the Hearing Restoration Program. That is a consortium of 15 labs working on it together. But the niche that we took and the approach that we chose was use the following idea. And it's a shared idea with a long-term collaborator of mine, Lionel Kahn from Tel Aviv University, who's a mathematician and a bioinformatician. We've been working together for more than 2 decades. And we reasoned many years ago, before single-cell RNA-seq and other techniques that allow us to break down the tissue to individual cells, that if we had the ability to take different cells in the ear that have different functions, or maybe the same cell at different developmental time points, and see what are all of the genes that are expressed in that cell or the different cells at those different, at the given time point, we would likely find that there are many common genes. There are many genes that any cell needs. All cells need mitochondria, all cells need protosomes, all cells need many different common functions. But only neurons need axons and dendrites. Only hair cells need stereocilia. So there are going to be some unique genes. Now, in biology, generally speaking, gene regulation happens primarily, but not only, through transcription factors. Transcription factors are proteins that will bind to a regulatory region, often upstream of a gene, and they tend to have a parking lot. They tend to have a short sequence that is recognizable. And they tend to regulate a program. So we reasoned if, for example, a hair cell is at the point where it's now developing its stereocilia, and we identified those 2,000 unique genes, maybe 300 of them are going to be related to a certain function, and maybe 80% or 50% of those will be regulated by a single transcription factor. So if we would look at those regulatory regions, we would be able to find an overrepresentation of a random sequence that we will then be able to go back and match up based on a database and find what is the regulator that governs this particular time point in biology for this particular cell type. So this is the concept of the next few stories that I'll tell you about now. There was a problem though, because the ear is really complex and it's very small and it has a lot of different cell types. And at the time, we didn't have techniques that could characterize one cell type at a time. But I was fortunate to be in the Department of Otolaryngology, and our department research was focused on actually cancer immunology. And instead of PCR machines in the lab, we had flow cytometers. So I realized that the most abundant resource was boxes of antibodies for cell surface markers. And I looked at one of the gene expression data sets I generated, and I realized that many of those cell surface markers are expressed in the ear. So we started just throwing these antibodies on inner ear tissues. This is a cross-section of a cochlea, and you can see here the hair cells in red. You can see that there's an antibody that separates epithelial and non-epithelial cells. And we identified additional antibodies that allowed us to break down the ear like a puzzle to different populations, that when we put them together, you can see here in the bottom right, we got different populations. Well, then we had a new problem. We wanted to generate the transcriptome of these cells, but the amounts of RNAs we were starting with were very, very small. And we were again fortunate because Lisa Sadwich and Luke Talman helped us a lot, both from the Department of from the Institute for Genome Sciences, and they've been working with us in every step of the way over the years, calibrating and changing protocols to make things work for us, because we always work on such small numbers of cells. In this initial study, we just took the epithelial versus non-epithelial cells, and we were able to identify a key regulator as the regulator of epithelial cell development in the ear. But then we became eager to figure out more about hair cells, and particularly the time point of immature hair cells, where they're there, but they're not really functional. Because when you take stem cells and you try to turn them into hair cells, you get hair cell-like cells, but they're not really functional and they're not fully mature. So for this, we combined this method that we developed with a mouse that expresses a green fluorescent protein in the hair cells. And you can see here the 4 rows of hair cells marked in green in the cochlea and then in the balance systems. And that allowed us to, using flow cytometry again, identify the hair cells, the supporting cells, and the rest of the cochlea, get them in tubes, and now we had another problem. The amounts were so small that even the current protocols modified were not good enough. And for this, Dr. Yang Sung from the IGS helped us, and she actually has a paper on that topic, how we were able to modify protocols to get reliable transcriptomes. And that allowed us to identify 2,000 genes that are uniquely expressed in hair cells at that time point. Many of them we, in collaboration with the University of Iowa, added to a sequencing platform where patients that have hearing loss and a mutation in a known deafness gene was not identified, these were added to, and some new genes were identified. But mainly what we were interested in was to find out, is there a short sequence upstream of the regulatory regions of these hair cell unique genes? And we found something. We found a sequence that I've never seen before in the ear with a very high p-value, not too far from the transcription start site. But the sequence was a match to a family of transcription factors that had many transcription factors in it, about 8. And about 5 of them were expressed in the ear. And it was going to be a very big investment to study these transcription factors at the biological level. So we tried to think of a different way to convince ourselves that this is real. And we reasoned that while the coding sequences, the sequences that code for exons, are highly conserved across species, the areas that are upstream between genes, what used to be called junk DNA, are really not, unless there's something very important functionally in them. And so we pulled those 2,000 genes in zebrafish and in human, just those regulatory regions, and we repeated the analysis, and we tried to see, are we going to be able to get something that is even a little bit similar? And we got the exact same sequence. And this indicated that this family of transcription factors is probably an evolutionarily conserved family of transcription factors regulating the terminal differentiation of hair cells, the sensory cells. So I mentioned there are several of them. We were very excited. Some of them, the mice die if we delete them from the mouse. So we generated conditional mice where we deleted these transcription factors just from the hair cells. So what you see here are hearing curves of mice that do not have either RFX1, 2, 3, or 7, the highest expressed RFXs in their hair cells. And here we're measuring the hearing at different frequencies. The black and the red, the black are the wild type, the red are the mutants at different ages. And what you see is that there is no difference. The hearing when we made all these mutant mice and the wild type and the mutants was exactly the same. Study section suggested that we cross RFX 3 and 7 because they had the highest level in the ear. We did that. That was a lot of work. And they had normal hearing. Well, in the pancreas, RFX 1 and 3 work together. So I still remember this day. They were 3 months old. I took them and did ABR. I was still a resident here at university. And the mice were completely deaf. We took this batch of mice, euthanized them, and ran with them by the next week to the scanning electron microscope. And this is what we saw. At 3 months, instead of having 3 rows of outer hair cells and a row of inner hair cells, the outer hair cells were completely gone. It was exciting for 10 minutes because we did this work at the time the mouse was just born. And this is supposed to be a terminal differentiation factor. It meant that we missed the party. So now we generated more mice. We got funding, and Dr. Milan joined my lab. She was working at the time with Dr. Ginsberg, that I'm excited to see here today. She was a postback in my lab, and now she's a pediatric GI fellow at university. And we started doing functional studies. We started from early on, and you can see here that both the wild type and the mutants, they internalize a vital dye through the transduction channel. We looked at markers of differentiation, and they were present. We looked ultrastructurally, and everything looked the same up to a week after life. Hearing in mice starts at postnatal day 13. Up to postnatal day 12, you can see 3 rows of outer hair cells, a row of inner hair cells. But 3 days only, the outer hair cells are gone. Robust, rapid loss of the sensory cells in the ear. And here's the question. What's happening? Is it a terminal differentiation factor and something is missing, and that's why the system's not working and notwithstanding the effort of hearing? Or are these some new survival factors that we can use in order to prevent hearing loss in more mature ears? To answer this question, what we needed to do is we needed to generate a whole new series of mice, and this is ongoing and new work in the lab that's unpublished, that grow to different time points. And only then we take those transcription factors away. So we already know that if we delete these transcription factors very early on, the hair cells died, the mice are deaf. We tried different time points with different Cre drivers, and there was no difference. If we took them away at 3 weeks, there was no problem with hearing. We took them away at 9 days, there was no problem with hearing. We took them away at P0, terminal differentiation, there was a hearing loss. This meant that the critical window is between the generation of the hair cells and about a week after the mice are born, the time of terminal differentiation. And these factors we're trying now to understand what are they regulating, both in the cochlea and in the vestibular system. And this is work that is carried out by Katie William from my lab. But hair cells, I told you, are divided into 2 groups. There are inner hair cells that are innervated, sensory, and there are outer hair cells. The outer hair cells, though, are very important because they fine-tune the sound, and they're also very sensitive. These are the primary cells that die with noise and aging. Even a single noise exposure causes their death. And they're incredibly difficult to differentiate from stem cells. When we take stem cells and we try to turn them into hair cells, we don't get outer hair cells. So we wanted to figure out what makes an outer hair cell an outer hair cell. What is that switch that happens? This is a project that was carried out by Dr. Maggie Matern that did her PhD thesis in my lab. And we were fortunate to talk with a colleague on campus, Mary Kay Lobo, who is a doctor at the Department of Anatomy and Neurobiology. And she told us about some mice she had where you can get the transcriptomes of cells of interest without having to dissociate the tissue. And that was important because outer hair cells would die if we dissociated the tissue. And so we did that, and we used similar techniques to what we've been using all along, bioinformatically, and we got 5 candidates. And we were at a conference, and a colleague of mine, Mike Bowl, from the UK, he's the head of an aging screen. And he came up and he said, you know, you guys do transcription factors. We identified a transcription factor in an aging screen that is uniquely expressed in outer hair cells. And the mice have their ears formed, but they're simply not functional. The outer hair cells are there, they're not functional. And he showed me staining for that transcription factor. It was expressed only in the nuclei and only in the outer hair cells. And it matched one of our top 5 candidates. So we looked at the mice, and indeed they had hearing loss, and the outer hair cells were not functional. But the question was, does this completely solve the puzzle? So to solve the puzzle and prove that this is true, we need to be able to take an inner hair cell and turn it into an outer hair cell, or at least turn off some of the programs of inner hair cells and turn on some of the programs of outer hair cells in the inner hair cell. To do this, what we did was we took mice that are just born, so the hair cells are immature, and we injected either a control virus or a virus with these transcription factors to the ear. The mice had a red fluorescent protein, so we'll be able, using flow cytometry and the core services, to pull out the hair cells. And then with the help, again, of Luke and Lisa, we did single-cell RNA sequencing to identify the transcriptomes of every single cell separately. The control mice provided us, for the first time, with really great markers for inner hair cells compared to outer hair cells at about a week after birth. And now we were able to ask, using the mice that got the virus, how are the inner hair cells that got the virus that we think is in charge of turning outer hair cells to outer hair cells different from normal inner hair cells? And what we found is as follows. You can see here on the left, the two left columns are always controls. The 2 right columns are always the injected transcription factor cells. Inner hair cell canonical genes that are high in inner hair cells, these bright pink, when we put the virus in, they were turned off. Outer hair cell canonical genes, when we put our virus in, were now turned on in the inner hair cells. And then we wanted to see if this is just something that's happening at the molecular level, at the gene level, or if this is something that's happening at the protein level. So Preston is the motor of the outer hair cells. It's a canonical marker of outer hair cells. It's expressed normally only in the 3 rows of outer hair cells. So we took mice and we did inject the virus to. On the right here are the controls. And 2 months later, we stained them for Preston. And you can see that in the injected mice, now we have Preston expression throughout the row of inner hair cells. And I can tell you that with a collaboration with Dr. Folenko, we were also able to show that they acquired electromotility, the canonical physical marker of outer hair cells. They did not completely turn into outer hair cells, though. I would say that there are probably 4 or 5 major programs, and we were able to turn on about 1 of them. The top of the cell remained the same. So there's still work to do, and that's good, because this is a project of Christopher Schultz in my lab, who's a graduate student. And last, I've been talking about hair cells all the time, but really, as the ear develops, the most common cell type is the cell type that's marked here in red. And these are the mesenchymal cells. They surround everything in the cochlea. And mutations in genes that are expressed in mesenchymal cells actually cause hearing loss in human. And because they surround also the nerve, a colleague of ours at Georgetown University, Tom Coate, was very interested in them. So we decided to collaborate, and Kevin Rose, who's a graduate student in my lab, decided to take on this project. And he's the ambassador for the mesenchymal cells in the cochlea. And we decided to focus on a transcription factor and figure out PAL304 that's uniquely expressed in these cells and mutations in it cause hearing loss in human. And the hearing loss that it causes in human is unique because these patients don't do well with cochlear implants. And when we looked at the mice and at the humans, we realized that there's too much going on in their ears. The cochlea are shorter, the nerves are not reaching the right direction, they're missing the endocochlear potential. And it didn't make sense that the mesenchymal cells had the same role everywhere in the cochlea. And we started thinking maybe these cells that we're calling mesenchymal cells, they're not just one population of cells. Maybe there are several different populations of cells, and we just call them mesenchymal cell because right now that's the marker that we have for them. Maybe there are several different types of cells with several different types of roles. So Kevin took mice that are a week old, he dissociated them, and he did again single cell RNA-seq. And he was indeed able to show that the mesenchymal cells are divided into 4 different populations with markers, very unique, robust markers for every population. And not only are they divided into 4 different populations, when he used antibodies to stain for these populations, he was able to show that each population was in a different area in the ear that marks a different structure of the ear as it develops. And not only that, when you looked at the wild type and the mutants, the genes that they regulated were completely different. And it's probably one of the first examples I can think of a transcription factor that regulates such divergent programs in one organ, but different and related cell types. So this is ongoing work and very exciting. These are the things we're doing for hair cell regeneration and trying to map out how the ear develops. But equally exciting, if not more, is how can we stop? How can we stop the damage that happens to the ear? And that's a great question. We thought about it a lot, and we tried to think what will be the most reasonable approach for this problem. Now, functional studies give us mechanistic cues to pathway that underlie noise-induced hearing loss and age-related hearing loss. And from the literature and years of studies from other people, we know that noise causes dysfunction of the sensory cells, marked here, and the supporting cells. The potential of the ear that comes from the side wall, the lateral wall, decreases. It's called the endocochlear potential, and we know that there's a decrease in potassium ion secretion. And then the neurons that connect to the inner hair cells, something very interesting happens with them. It turns out from research of just the past few years that we know of markers that divide them into 3 different groups. And the 3 groups are distinct. Type A, 1A, are very resilient. And even relatively loud noise exposures don't bother them. Type 1C are extremely noise sensitive, and even noise exposures that don't result in a change in hearing threshold result in their loss. And it's thought that type 1C, because they have a higher threshold and lower spontaneous rate, are incredibly important for hearing the presence of noise. And they've been shown to retract throughout the cochlea as we age. So it's thought that the reason when we sit with our parents at a restaurant, and even if our parents don't have hearing loss, they have a hard time following the conversation if there's background noise, it's because the 1C retracted. And there's a lot of pharma out trying to figure out how can we get the 1C to come back to the inner hair cells. And then there is an activation of the immune response. So what we see here is that every part of the cochlea has something completely different happening in it. If we take the cochleas and we mush them together and we do RNA-seq, we're not going to find out a lot. Or this was done already, and mainly what people found was activation of the immune system. So we set out to build a map of the molecular changes that happen after noise exposure in order to do rational drug design. And for this, we put together our largest collaboration. Our team here led the work. A lot of the data sharing was led by our team members here at the IGS. Ran Elkan at Tel Aviv led the bioinformatics. We had pharma collaborate with us and contribute data they generated and we analyzed, and a group that focuses on the immune system and the Karolinska. And all different sites took mice that had normal hearing and exposed them to noise that was deafening across all frequencies. And then every site collected the cell type of interest. We used the ribotag mice to not dissociate the ear and figure out what's happening in hair cells and supporting cells. Decibel did the lateral wall and ganglia, and Karolinska, the immune system. And then we started looking at what's happening. And this is a very long story that was just published this month in Cell Reports. So I'm just going to give you two short stories from it. Of course, we found that there are things that happen throughout the cochlea, or at least throughout the sensory epithelium. Increase in cell death, increase in immune response, decrease in some genes that are not specific to a cell type. We also found some phenomena that are unique, you can see here, to outer hair cells, genes that go down only in the outer hair cells, or go up only in the outer hair cells, or go down only in the supporting cells, and up or up only the supporting cells. I want to tell you, though, about the neurons, because I really am fascinated by the neurons. And it would be great to be able to prevent that neuronal retraction. And going into it, we were hoping that we'll be able to find the different populations, and that we'll find that type 1C neurons that retract turn on a program that's really bad. And then we're going to cross it with a drug database, and we're going to find which drug to give to prevent this retraction. Things could not be more different. If you look at the top line here, type 1C neurons have absolutely nothing happening in them. Very few genes go up and very few genes go down. Type 1A neurons, though, have a very large number of genes go up and a very large number of genes go down. This suggested that maybe type 1A are doing something to protect themselves. Went ahead and tried to find, can we see what regulates this molecular response? And we found that 125 of the 228 genes that go up in the 1A neurons that are resilient are regulated by a single transcription factor, or at least bioinformatically predicted to be so. It's a family of the ATF transcription factors that are also upregulated and are known to be negative regulators of cell death and neuronal death. We wanted to make sure this is actually happening. So we did stainings in cross-section. This is work that was done by Erica Lipford from my lab, who's now a medical student in the school. And so you can see that ATF3 and ATF4 are upregulated strongly in the ganglia. You can see that their canonical targets are also upregulated after noise. We're further able to show that this upregulation is not throughout the ganglia, but only in select areas that do not overlap with a marker for type 1B and 1C neurons, which shows that this is unique to the 1A neurons that are noise resilient. And now we're working with Dr. Shear and Dr. McCarthy to actually do this induction in the type 1C neurons. We also saw a universal response to noise that was related to immune genes, and we found their regulators. That's another line of research. And finally, we looked for drugs that can counter this response. And basically, we took the gene changes that we found, we crossed with a drug database, and we looked for responses that will be opposite. And the top candidate drug came out as metformin. And that's really interesting. Some of you might know that metformin is currently being investigated as an anti-aging drug. Metformin affects the mevalonic acid pathway that we know has a negative effect on hearing when it's activated. We also know that noise activates mitochondria complex 1, and that inhibits AMPK, that results in activation of the mevalonic pathway. Metformin inhibit several of the mitochondria complex 1 components, inhibiting the mevalonic acid pathway. And it's really neat because it's an old drug. It's not under patent. It's cheap. It's well tolerated. And in people that don't have diabetes, it does not cause hypoglycemia. We had clues to the fact that this is probably important because Lisa Cunningham from the NIDCT, she's shown that statins that function on the same pathway, to inhibit the same pathway, can prevent cisplatin ototoxicity in mice. And in fact, we're now a site in the study that Dr. Cunningham is leading, and we're collaborating with the Cancer Center and Dr. Mera to see if we can prevent the cisplatin-induced ototoxicity in our head and neck patients that are getting cisplatin by treating them with statins. So Dr. Katie Kennedy from our department, a resident that rotated under the guidance of Benjamin Schuster from my lab, decided to take this project as her main project and see if she gives mice metformin in their water, can she prevent or decrease the noise-induced hearing loss? And the graphs here on the right, and this is a study that is still ongoing. This is our first cohort. We need to repeat it multiple times. We need to extend it also to females. But the study here shows great promise. And you see that the threshold shifts both 1 day out and a week out, and in particular in the higher frequencies that are the frequencies that are affected with aging, are significantly smaller in the mice that got the metformin. This project is now carried out by Dr. Amanipur, a postdoc in the lab, and Benjamin Schuster. So you might be wondering, we're doing a lot of gene expression studies, and we're generating valuable data, and then other people in the field are generating similar data. How are all these data become accessible to everybody? How can we make it that all this gene expression data and products such as those generated by the Institute for Genome Sciences and similar institutes across the country become as accessible as reading a manuscript through PubMed? It will be unfair that such valuable data generated all the time would require always a middle person, such as a bioinformatician. There are not enough of those. So we wanted to figure that out, and we came up with the idea of the gear. And I still remember presenting the gear to Dean Reese maybe 7 years ago, and was barely an idea. And for this, we assembled an amazing team led by Anoop Mahorkar from the IGS, who is my trusted colleague and partner for many years there, and Joshua Orbis, our lead engineer. They're joined by Sean Atkins and additional engineers from College Park. And in a nutshell, you can take any gene, put it in our search box, turns data into presentations that are intuitive. And you can choose if you want to see them as graphs, as cartoons, as dot plots. You can customize it. For the ear field, we curate everything. We have hundreds of curated data sets, so many data sets that people don't even know that exist, organized in thematic pages by topic. And then it turned into the main go-to for gene expression in the field with thousands of visits every month. People can learn about the gene, the annotation. They can find out if a gene causes hearing loss in human or in mouse. And they can learn more. They can see the publication it came from. They can, just by playing with the platform, look at differential gene expression, or for single cell data that is a newer modality that's incredibly challenging to download and analyze. They can actually get access to the data and work with it seamlessly. There's a dataset manager. You can look for your datasets of interest. It's a very nice platform, and was recently published in Nature Methods this year. This platform, we realized, has more potential, and we looked for additional collaborators on campus. And we found people with interest in the neuroscience, including Dr. Seth Amit and Brian Herb, also from the IGS, and we were able to repurpose it for the neuroscience community. And this actually became one of the official visualization environments for the BRAIN initiative that just this month published a series of papers in Nature, the motor cortex single cell transcriptome. And every paper there in their figure legends actually links to our platform and allows people to not only read the paper, but seamlessly access the data and use it for their own personal interests and discovery. For lack of time and not for lack of importance, I'll just mention two other collaborations that we have on campus without too much data to present today, though they have a lot. In our studies, another thing that we found was that males compared to females are more susceptible to noise trauma. We reached out to the animal facility to ask who works on sex differences on campus, and we found that this is a major site to study sex differences. We partnered with Dr. Jessica Mung, and we've got a whole series of papers on this topic, and we're now working on figuring out how can we repurpose this, and at least in females, prevent noise and age-induced hearing loss using the estrogen signaling pathway. And this is really work led by Benjamin Schuster from my lab, a postbac that his bigger paper just almost got accepted this week, hopefully next week. And then we recently built, 4 years ago, the MSICE, the Maryland Cochlear Implant Center of Excellence, that bridges between College Park and University of Maryland, Baltimore, to really capitalize on the research capabilities in College Park and cochlear implants and benefit our patient population here. And we're collaborating with Dr. Rezudin and Dr. Ahmed here, amongst other things, to learn about the molecular basis of hearing loss in our African-American population and correlations between implant performance and genetic markers. So with this, I'm done with data. And before I continue to the regular acknowledgements, I wanted to give special thanks to 4 people that, in my opinion, made a huge difference to my success and well-being on campus. And the first thanks is to Dean E. Albert Reese for establishing a culture where research is an important value for both clinicians and scientists, and promoting the clinician-scientist track and enhancing the school research infrastructure. Thank you. The second thank you is to my department chairs, past and present. It's not an easy task to support the clinician scientist. It's an uphill battle, particularly in the surgical department. I want to thank Dr. Strom and Dr. Taylor for valuing the role of clinician scientists, protecting the time for research, and supporting the mission. And last but not least, I'd like to thank Dr. Frazier, that when I was a third-year resident, took me under a wing and accepted me as a mentee, and later a faculty mentee, made me a member of the IGS, made the IGS my research family here, and supported my professional development. And with this, I just want to thank my team who's here, my collaborators, our funding resources, collaborators that did not have a chance to thank today. I will mention that we do have an opening for a postdoctoral fellow, and I'm happy to take questions.
Thank you so much, Dr. Hertzano, and congratulations. So we are now going to move into the Q&A portion of our event. So anyone in person, if you have a question, go ahead and raise your hand and we'll bring the microphone to you. And anyone watching virtually, if you have a question, to put that in the chat window and we'll relay it to Dr. Hertzano.
Circling back to the beginning of your talk, so EpCAM and integrin alpha 6. So did you find out what they're important for? Is it positioning of the cells, or you never went back to study that?
So for EpCAM, which is also called CD326, we got lucky. We were stuck in a snowstorm in Disneyland in the meeting, and I was still a resident, and I tried to find someone I could brainstorm with. And So I decided to brainstorm with the scientific director of the NIDCD at the time, Dr. Griffith. And he looked at me when I said EPCAM, and he said, which is called, and he said, you know, ZEB1, which is the regulator we identified, he had a point mutation in a non-coding area. And they made all these mutants, and they couldn't figure out what's the function. So we got these mice when we came back, and we did gene expression on epithelial versus non-epithelial in these mutant mice, and we found that it indeed regulated the epithelial domain. What it actually does, we don't know. Nobody tried to actually completely delete them in the sensory epithelium. We do know that the hair cells downregulate EPCAM a little bit, but that's as far as I can go.
Ronna, that was spectacular. That's better than my private presentation. This is really the whole story. I got a question though. Excuse me. I was fascinated by the fact that the outer hair cells, when you used stem cells, did not produce them in totality. And from a developmental biology standpoint, most organs are not produced either by the same cell or at the same time. And they have varying levels of susceptibility to injury and to formation. So I wonder whether you had tried multiple different stem cells or multiple stem cells simultaneously to see whether or not you would have been able to induce their function, not by a single set of stem cells, but by multiple. And secondly, whether or not you mentioned that the various hair cells some were susceptible to sound, to noise, and some were not, and whether or not they were correlated with potential embryogenesis development, because we know that in other settings, depending on when, let's say the heart or the neural tube, they may be much more susceptible based on when they were formed in utero.
Great questions. Depending on the stem cell, we don't do stem cell research. We hope at one point to collaborate with Dr. Zelsman to do stem cell research for the ear, because her background is from there too. But groups that have done work with stem cell research, regular stem cells or induced pluripotent stem cells really don't make it too far. In organoid models, so 3D organoids, there are two main approaches. One is with morphogenes that are added or with serial transcription factors. And there they make it a little bit further, but most of the cells are still more inner hair cell-like. When you take, instead of just regular stem cells, you take cells from the organ of hearing, so supporting cells that are specifically LGR5 positive, and now you generate an organoid, you can get more outer hair cell-like cells that will express some of these markers. But you're not going to regenerate an organ by taking the organ and mincing and doing that function. One of the cool things, though, are that, right, and even when you take mature ears, it's still hard, though I think we're getting to the point that people will be able to take supporting cells in mature ears and turn them into more outer hair cell-like. But just in the last 2 months, a group in China published a very nice study, and they showed that when they take IKZF2, they can actually turn adult supporting cells into more outer hair cell-like. So that for us was really nice because it showed the value, right, of publishing the data and sharing it. So I think that exactly like you say, it's going to be finding the right cell to capitalize on, finding the right combination of transcription factors, finding ways to not just turn them on, but also turn them off. So transiently activate them. And that's tricky.
We have about 5 minutes left. We have about 5 minutes left in our event today, so we have time for one more quick question, I think. Well, from the virtual audience, Frida Korenbrot says, thank you, Professor Hertzano, and really great talk.
Thank you.
Well, if we don't have any other questions for Dr. Hertzano, I think that concludes our event for today. So congratulations. Let's give her another round of applause.